this presentation is a short one. We don't have much time. So without further ado, I will try to go into the more specific aspect of the presentation, which really is about the, as we were requested, trying to show what are the new tools that we have been using very recently at IRRI to progress our genotyping effort. So I will be focusing on two new tools that we are using already and advancing. Some of you are very familiar with that because you are specialists, the SNPs and single nucleotide polymorphism, and some tools that are more recent and less used, relying on machine learning. Of course, now everything is artificial intelligence, and we look at that a little bit more. So first, I think it would be interesting for all of you to have a brief overview of what the IRRI Gene Bank is all about. You probably all have heard that it is the oldest in the CGIAR. Also in terms of accession for a single crop with more than 142,000 accessions. Diversity is also very important with more 130 country contributions, and also a very broad coverage in terms of diversity. Also in terms of use, not only conservation, we are proud to say that we are distributing samples regularly and steadily with a good growth in these aspects as well. So IRRI Gene Bank is probably one of the pillars in the CGIAR and beyond to actually make sure that we disseminate this culture of conservation and use. And part of the talk is also about what is being done in terms of research to actually improve the knowledge that we have about what we are the custodian of. So here, very rapidly, Charlotte is here, so she knows that better than anyone. The IRRI Gene Bank was the first one to receive this funding in perpetuity from the Crop Trust. We have gone through the second round of that. Here you see the Filipino president with me for his visit to the Gene Bank, and trying to really better understand what could be the benefits for the Filipino rice sector. So here is the most technical part. So as you all know, the big problem with the Gene Bank at IRRI, for example, I guess for others, is that we are not yet using the full potential of the genetic diversity that we have, roughly speaking 7%. Part of the problem comes from the fact that we don't really know what we have. And we don't really know what we have because we are not being able to identify, characterize the subpopulations and therefore the traits that could be interesting to help us tackle the challenges that we are trying to tackle. And so for a long time we have been using what you know is the morphology characterization with its also well-known limitations. So that was abandoned at IRRI. We no longer do that. We have for a while relied on the SSR, which stands, if I recall correctly, for isozyme, a simple sequence repeat. So this is something that we have also abandoned, going beyond to now rely primarily on the high throughput method called the SNP that I was mentioning before, a DNA-based marker, and that is now the way we do business. So what is very important to note is this is all the story of partnership within the CGIAR, but also outside the CGIAR. And so these genotyping, we don't do it ourselves. We use, as you can expect, outside services, providers for this genotyping work. And we have been working regularly with a company based in Australia that is called Intertech. So the example I wanted to use is this ongoing effort to actually better characterize sativa. So the species that is one of the two main ones. And so for that, we have used a DNA sequence, coming from a data set of 3,000 genomes, and we studied out of that millions of these SNPs and identified a minimum set of SNPs, roughly 40, that could explain the maximum variation that I was referring before in the subpopulation, so trying to understand this diversity. And of course, we went through a validation process, and we came down to 30 SNPs that were eventually identified. And these are now available as CASPs, you know, this method that is employed to genotype for SNPs. So all that to say that we find, and this is also published, that this is a very reliable method with a high degree of repeatability, which is what we all want to see. So others could do and check, and it is also, of course, documented. So also another thing is, sometimes I am asked the question, but how costly is that? So the cost have reduced considerably. And now, for example, just to give you an idea, when we send a sample to Intertech, it is roughly an average cost of $4 per sample. So it is a lot cheaper than what it used to be. And with a small number of these samples, we can identify subtypes and subpopulation quite reliably. And so this, I think, is really the key message. So this is just one of these examples where we took this chip and we genotyped more than 60,000, as you can see on the slides, 50,502 accessions from the gene bank. And through this information, we have been able to generate this graph showing, you know, what is the composition. And of course, you can see that it is largely dominated by indica, which is not a surprise, which is the most common, you know, variety that is being used. But this could help us over time and gradually to better characterize the whole gene bank, the 132 plus accessions. So in this other slide, and so using also a different species of rice, and at IRRI we are very much focused on these wild species of rice to actually try to find these attributes that we are interested in when we talk about climate change, when we talk about resilience, when we talk about some characteristics or traits that could help us to address nutrition, for example. We go back to these wild pieces and we look at what could be identified. And so we have devised with the same method another SNP panel with over 24 validated NSPs. And here we will actually, this is ongoing work and it will be public and published soon. Everyone and anyone can use this set. It is available. And here, what we show on this graph is the performance of this test when it is tested on a sample of gene bank accessions. So basically, you see here the taxonomy, and you can go down to the variety level and understand, you know, what are the subgroups, subpopulations that we have captured out of these SNPs. For those who are interested, I can share the slides, but it's quite you know, particular. This is now the second example of what we are doing with machine learning, artificial intelligence, totally different method, of course. Here, we basically rely on comparison in terms of image. So we are using seed images. So we take one seed, for example, and we compare to what we have in the gene bank. And so this is work that we have done during the COVID lockdown. That was quite tough in the Philippines. And so when scientists had nothing better to do, they did that. And so this really helped us to accurately identify 85,000 varieties. And so as part of a gradual process that is, as I was saying, ongoing,
we hope that we could identify and characterize the 132,000 accessions. So the issue here, because nothing is perfect, and that is also the question that we have not yet answered, is answered at IRRI, is are the seeds we have the original one? And that is difficult to tell. So therefore, we are also doing some duplication work and duplication of efforts, because we know that we have duplicates, and we know that some are not original. The problem is that we don't know which ones. And so, we are doing a better job, but we are not yet there. So also to talk about partnerships, we do that in collaboration with Bosch Germany, but the India-based branch. And so this is also something that all the credits for everything I'm saying goes to Venu in general, but this in particular, because the mad scientist who was doing that during COVID was him. And so this is really something that we believe is really promising. As far as I know, we are the only ones doing that in the CG. But of course, everybody could do it, because while it is top science, it is actually not rocket science for those who know how to do it. So, so we believe that there is also a huge potential for partnership within the CGIAR, and make sure that we could try and test this in other gene banks. Well, that's it. I will stop here. I was, I think, 10 minutes. I'm not too long, and I will finish there. I'm happy to take questions if I could. Thank you very much.